art in the most fundamental way.
So I've been thinking a lot about what is natural, and we haven't really probed that subject matter that much. Susanna touched upon it a little bit in her talk yesterday, but I want to talk about the natural, what is synthetic, and what is post or transnatural vis-a-vis life. So ostensibly, we've been changing nature since the dawn of civilization. It's important to unpack the conditions for knowing what we know, the how and the why of what we know, with respect to natural versus synthetic. For example, what is agriculture if not bioengineering? I'm particularly interested in mutations, which happen all the time in nature. We are born from mutations. Genes move laterally between species, and so we can look towards a discipline like synthetic biology, which is a young field growing with momentum, and it's an attempt at designing biology rather than just to study it. It's related to genetic engineering, and genetic engineering is an example of something that would lead to transferring something like fish genes into fruit so that we can make cold-resistant fruit and we can have strawberries in January, something like that. But that's also under the framework of artificial intelligence and somewhat digital technology for the purposes of this summit, I guess. I don't have a vast array of slides, so just kind of randomly just go somewhere. So I'm specifically interested in the ethical issues that synthetic biology has given rise to about the origins of life and how it's dismantling our thinking on evolution. Synthetic biologists think evolution is baroque, it's error-prone, and it's difficult to decipher. And what constitutes a species? Transgenic technology is messing with the category of species. So again, I have to ask, what is natural? How do we manage the ownership of life's materials? What responsibilities does designing biology carry? And should that design respond to the world and reflect the values of it, especially at a time when we know we can now see our own species extinction on the horizon? So in 2015, okay. There we go. In 2015, I traveled to the Brazilian Amazon, primarily for its reputed vastness, its promise of innumerable biological specimens. In short, for the ineffable sublime. And I went there to make a 3D video film that premiered with an exhibition in Kassel that I did with Susanna. And so I was interested in this mythology like anybody else about the Amazon, and I was interested in what it means for humanism, if everything can be connected and that no distinctions are made. But mostly I wanted to re-examine the boundaries that we have placed between the natural and the unnatural. And I felt that the Brazilian Amazon was a pretty good model to examine. So again, we can look to synthetic biology, transnaturalism, posthumanism, to think about multiple forms of consciousness, like the cosmologies of indigenous peoples from the Amazon region. And these people, they follow a non-anthropocentric and non-hierarchical thinking about life, which is sort of described by an influential anthropologist that I was reading when I went on this trip and when I was making this film, who is Eduardo Viveiros de Castro, a Brazilian anthropologist. And he has sort of coined this term multinatural perspectivism. And it's ostensibly just taking into account that there are lots of different kinds of consciousnesses, and that maybe we shouldn't have a kind of hierarchical relationship where humans are on the top and other life forms are at the bottom. So, for instance, in my film, The Flavor Genome, that's the film title. Nature is not conceived in absolute terms, but it's assembled from a multitude of perspectives and perceptions. The film depicts a fluid mutation of species, at the same time showing how biology can no longer be separated from its narrative and its biography. So, to quote a line from the film, humanity still has not successfully metabolized the imperialist cultural pollution, the planetary despoliation, the vanished indigenous civilizations, key elements that comprise the flavor profile of the tropics. So, I guess this film is an attempt to explore the Amazon, but also to raise these questions of how perceptions can be changed, but also how sensory experiences can allow for a different understanding of perception and consciousness. So taking these new sensory discoveries as its point of departure, the flavor genome, the film, imagines a chemical synthesis of plant and animal forms through an embodied cognition. And so far I've been talking about animals and plants, but to further that idea, my work embraces non-human persons by straddling synthetic biology and companion species. I look for the companionship of bacteria in our biota. Wait, this is really, sorry about that. Okay, let me go back to a bacteria still. So in order to emphasize the cyclical, the recursive, and the metabolic. So biofiction attends to these mysterious cues. It notes the fundamental strangeness of cellular self-assembly, that which confounds entropy by giving a common energy to living things. And in a sense, that's been stigmatized as pagan or savage, the view of living entities as co-subjective with us. It's gaining ground. Life is being reshuffled and aided by synthetic biology, etc. Artists should play an important role in that reshuffling. We are entangled with objects, tens of thousands of species of bacteria in a given human's biota. We can think of theories of entanglement. I'm thinking about Karen Barad and the interconnectedness of all of us. And bacteria is the workhorse of synthetic biology, which has been a collaborator subject in my work for the past few years now. So before I get into how I've applied the bacteria, I just want to give a little context. I've been adopting the term biofiction when talking about my work. It's a term coined by my colleague Carolyn Jones, a scholar at MIT, where I did a residency in 2015. And biofiction for me means that which fuses the writing of life with the study of life, which also now includes an embrace of non-human persons. And non-human persons is a concept used by indigenous people. That's what I've borrowed. And all life is a person. So there's a plant person, an animal person, a human person, with what this Hungarian biosemiologist Jakob van Uexküll refers to as, every living thing has an Umwelt and ostensibly a surround world. So again, to reiterate, humans aren't exactly at the top of this hierarchy of life. And if we imbue a tick or a spider with the signals that might motivate her, she becomes a person with a world and a worldview. She is stimulated and perceived by a subject. So with that in mind, let's examine the materiality of this biofiction. I made an exhibition in 2017 at the Guggenheim Museum in New York City. For this exhibition, I worked with a team of structural biologists, forensic chemists, and perfumers, as well as another artist, to create an installation in which natural and technological forces appear as surging, unruly forms that are nonetheless clinically contained. So visitors, at their first encounter, there's an entryway, such as this, or a kind of holding pen, where insect canisters emit a scent. And I just want to maybe give a little backstory towards this. When I was preparing for the exhibition, Donald Trump had just become elected, and I was, like everyone else, just very distraught, flummoxed, and I didn't really know how to respond in an art context. And I was really thrown off. I didn't really know what my role was in this kind of state of crisis or something. So he was threatening to implement this ban on Muslims and trying to essentially, kick people out of Muslim backgrounds out of the country. And so I think that this was this kind of loose trace response, this sense of like a holding pen, this crowded, narrow corridor, and that people would just sort of have to wait in order to enter the exhibition. So I've consistently sought to generate a sensory immersion that challenges the ocular-centric experience of art. And scent is a recurring component in my work. I use it sculpturally, linguistically, and to tap into memory, intangibility, synesthesia, and a kind of presence through absence. So when I activate smell, it's not a substitute, rather it's an extension. I don't evoke scent where there is an absence of something to look at, but when I want to extend the senses, like rings of vision or olfactory, all triggers creating ellipses in consciousness. So with that in mind, I want to examine our senses and how they are shaped through perception. And that, I mean, I don't want to get too much into, the sort of finer points of olfaction, but smell is pre-verbal. And yet we can take the real smell of something and combine its functionality with the stimulus of its smell and unlock semiotic codes. So what you can see here are these insect canisters. And as I mentioned, I worked with a forensic chemist and a perfumer and another artist to create a smell. And I guess I should just sort of give you a little bit more of the biofiction context. Initially, when as I said, I kind of got a little bit, I wouldn't say derailed, but it was a moment of pause. I had a different idea, and I went to these structural biologists up at a laboratory in Columbia in New York, and I proposed a project. I asked them, well, can you help me create a drug? Maybe even if it's fictional, a drug that would allow a human being to experience the perception of another living organism. That could be a coral reef, a pink dolphin, or another human being. And they humored me for a while, and before they told me the inevitable that it's biologically impossible to do that, because in order for me to experience the perception of a coral reef, I'd have to remold my brain to the brain of a coral reef or a pink dolphin, and I would have to change all the neural networks, and we just don't have the technology to do that yet. And so the way that I was able to just sort of, I guess, move along with that narrative was that I decided I wanted to create a hybrid consciousness through olfaction, through chemical compounds. So I took sweat samples of Asian American females, and I took a chemical analysis reading of some carpenter ants. And so I felt that, well, if I can't literally experience the consciousness of a carpenter ant, perhaps if I combine their compounds together with the compounds emanating from the sweat of these human females, that would activate the biofiction this rubric that I've been working with for the past few years. So these canisters are emitting this scent, and the idea is that you would take it in through your, smell the fragrance, and that somehow that would activate a biofictional drug, and that you would be endowed with the perception of this hybrid consciousness, hybrid organism. And just, I'll just pepper a little bit of things. It's commonly believed that humans have a poor sense of smell compared to other mammalian species. A lot of people come up to me and say, I can't smell anything, and and I think that probably they're telling the truth, but it's kind of like playing the piano. You just have to practice it, and that's what perfume school is. You have to identify maybe a 1,000 different materials through your sense of smell, and you would just, like an instrument, just learn how to be more virtuosic with your instrument, like your nose. So I think I don't accept that any of us can't smell anything. I also think it's a kind of conditioning that we have, that in most public spaces, especially in New York City, we really, really try not to smell anything at all. So, and there are, legitimate reasons for that. But so this idea of humans having a poor sense of smell, it derives not from empirical studies of human olfaction, but from a famous 19th century anatomist hypothesis that the evolution of human free will required a reduction in the proportional size of the brain's olfactory bulb. The human olfactory bulb is actually quite large in absolute terms and contains a similar number of neurons to that of other mammals. Moreover, humans have excellent olfactory abilities. We can detect and discriminate an extraordinary range of odors, and we just are more sensitive than that. So this ties into what I've been investigating in my work for the last couple of years, and continue to do so, about how the senses are conditioned socially and how our perception is conditioned socially. So that, the ocular, our sense of sight and vision, is associated and attributable to the masculine because it represents discovery, knowledge, moving forward. And that's also a kind of a result of, maybe a skewed idea of evolution, when we got up off the ground and we became erect bodies and we started to privilege eyesight. Whereas then olfaction got hurtled back into the Middle Ages even further, because it became associated with that which was inscrutable, mysterious, and entirely subjective. And then what would you call that? You would ascribe that to the feminine, which I reject. So this work was called Immigrant Caucus, and it combines chemical compounds derived, as I said, Asian American women and carpenter ants. And so you walk in, you're inoculated with this biofictional drug, and you see this sort of museological architecture of a diorama. The gallery's central space features 2 opposing dioramas, each providing a view into a self-contained biosphere. The first is lined with tiles that hold a gelatinous substance called agar, which is made of seaweed. And I cultivated various strains of bacteria sampled from sites within Manhattan's Chinatown and Koreatown neighborhoods. So this living composition also blooms across several sculptures, as you can see, as if an invasive life force has overrun the environment, kind of my sci-fi biofiction. By using microbial materials, I wish to investigate paranoia around contagion and hygiene, which is a prominent preoccupation in our society. But but it functions in so many multiple ways. On the one hand, it's creating a living material representing the specific area of Manhattan in New York City. You could even regard it as a portrait, if you will. But also, because bacteria can exist for hundreds of years in a Verbier air duct or something, you know, you're actually also then incorporating history, but we're talking about bacteria's metabolism and entropy by germinating a vast and violently bacteriological tableau. So, wow, you know what? I really cut this talk down. I thought we only had 20 minutes, but I can extemporize. So, but also the bacteria functions as a time-based work, and the microbial sculpture renders visible the source of our unease while foregrounding the politics and subjectivities of smell and touch and its impact on our empathic understanding of each other. Just to give you a little background on when I first started intentionally working with bacteria. I mean, I've worked with bacteria, but not intentionally as a material. But I think, again, I mean, I am somebody who exists in the world. I respond to the world. I communicate in the world. And in New York City, I think it was around 2000, I want to say 14, there was this whole Ebola virus contagion, freak out, paranoia, and nobody wanted to go in these sort of crowded spaces. Nobody wanted to take the subway. I took the subway, and no one wanted to touch the subway pole. And people were genuinely frightened because there were a couple of doctors who had got struck with the virus, and they were quarantined. And so these were, you know, just the 24-hour news cycle inundated with this image of the nurse who was quarantined in this tent. And so I thought that was a very palpable mood to think about making art. So I thought about contagion and this sense of hygiene and our discomfort around disease, and I thought that's really, you know, a formidable material to start working with. But also, there's a visitor to, let's say, an exhibition that contains, you know, a smell component or bacteria. You're not a passive visitor. There exists a symbiotic relationship between the work and the visitor, because you actually have to inhale the chemicals inside your orifices through your nose and the pores of a person. And in that sense, the person contributes to the biota tableau in the diorama. So throughout the run of the exhibition, it was maybe like for, how long was that, 3 months or something? It started out with a few spores, a few blooms, and then progressively the exhibition, I mean, the diorama just started to bloom. And, but I mean, on a formal level, it was really interesting. It's kind of like, I don't know, trying to paint with a very uncooperative, unstable, unpredictable pigment and, you know, shading and all of that. So I guess technically speaking, there were 225 tiles, and you're looking at basically a very, very sophisticated refrigeration unit, and it's a, there was a whole sort of walk-in refrigerator that there's all this sort of walkway in the back that you could enter and sort of access the tiles, and only myself and the biologists were able to handle the bacteria. So, but anyway, it kept transforming. On the other side was another companion diorama, and it houses a colony of ants. And Okay, yes, perfect. Insects, yes? No, I'm gonna wrap this up right now. Uh, Insects interest me because of their intricate division of labor and matriarchal social structure, as well as their sophisticated olfactory system that guides their behavior. The ants navigate a network of pathways that are reflected infinitely across mirrored surfaces, evoking a massive data processing unit in which their industrious movement embodies the flow of information. The colony is exposed to the same hybrid scent that fills the corridor leading into the gallery, creating the possibility of a shared psychic experience between ant and human. Ants in this way are also biopolitical agents, much like the bacteria are. So in closing, I started with a pure fiction, a kind of a wish, and in a way, scent is a wish and a fiction.